In this video, we're going to talk about the pentose phosphate pathway, also called the hexose monophosphate shunt. Pentose phosphate pathway occurs in the cytosol of all cells, but its activity is higher in cells with specific demands for biosynthetic or oxidative stress protection needs. Its major purpose is in producing NADPH, which is a crucial reducing power used in biosynthetic processes like fatty acid and cholesterol synthesis, which is important for tissues like the liver, adipose, adrenal glands, gonads, mammary glands. It's also used for maintaining reduced glutathione to protect against reactive oxygen species, which is very important in cells in direct contact with oxygen, like your red blood cells and the cells of your eyes. We also get ribose 5-phosphate, which is important in DNA and RNA synthesis, and for things like ATP, NADH, and coenzyme A, which are very important for rapidly dividing cells. Some of those rapidly dividing cells are good, like bone marrow cells or skin cells, any of your tissues that have high cell turnover. Other rapidly dividing cells aren't so good, like tumor cells. For NADPH, don't confuse it with NADH. They are structurally different and have different functions. NADH is primarily involved in catabolic reactions, which is your breakdowns like glycolysis, TCA, oxidative phosphorylation, while NADPH is primarily involved in anabolic reactions. The high energy form is NADPH, which carries the electrons and donates them in reductive reactions, while the low energy oxidized form is NADP+. What NADPH does is it helps other molecules be reduced, being oxidized itself at the same time. So the goal of this pathway, one of the goals, is to have NADP+ accept the electrons to give us NADPH. Another product is the formation of ribose 5-phosphate, which is important in nucleotide and nucleic acid synthesis. It consists of two phases: the non-reversible oxidative, where we get our NADPH, and the reversible non-oxidative phase, where we get the interconversion of many different numbered carbon sugars that can serve as intermediates for glycolysis or gluconeogenesis. The oxidative phase starts with glucose 6-phosphate, and with every one of those, we get 2 NADPH, 1 ribulose 5-phosphate, and a released CO2. We start the first phase with glucose 6-phosphate, which is also part of glycolysis and gluconeogenesis. The first enzyme is glucose 6-phosphate dehydrogenase, the slowest and rate-limiting step. When we see dehydrogenases, remember to think redox reaction. So we'll have our oxidizing agent, NADP+, accept electrons and proton to form NADPH, the reduced form, and also releasing this hydrogen. We'll have a magnesium to help support this reaction, while our glucose 6-phosphate is oxidized to form 6-phosphogluconolactone. So this first step is the oxidation of glucose 6-phosphate to get that first NADPH. For the enzyme here, glucose 6-phosphate dehydrogenase, there is a known deficiency that can result in people getting sick if they eat certain foods or if they take certain medications. But we'll get to that later. Next is a hydrolysis reaction of 6-phosphogluconolactone. So we'll have our water come in. And this is done by the enzyme 6-phosphogluconolactonase, or just lactonase, where the cyclic lactone is hydrolyzed to form the open chain 6-phosphogluconate. We'll have another dehydrogenase, 6-phosphogluconate dehydrogenase, that catalyzes the oxidative decarboxylation of this 6-phosphogluconate. So we're also losing a CO2 in this step. We'll again have our NADP plus accept the electrons, meaning it's reduced to NADPH, and the 6-phosphogluconate will be both oxidized and decarboxylated. Our magnesium will support the reaction as a cofactor, and this will get us to ribulose 5-phosphate, 5-carbon sugar. We now have our 2-NADPH and our ribulose 5-phosphate. To prevent any confusion, some textbooks will say that this is the end of phase 1, while others will say it's when we get to ribose 5-phosphate. This last reaction where we get the ribulose 5-phosphate is the last oxidative reaction in the pathway. However, because it rapidly isomerizes to ribose 5-phosphate, some will extend phase 1 to include the reaction where ribose 5-phosphate is made. If you see that difference between what your professor says and what the textbooks are saying, or what some other source online is saying, just do whatever person who's going to mark your exam tells you to do. But if you do see resources that show the line between the two phases differently, that's why. Phase 2 is the non-oxidative phase, and this first step is where we convert our ribulose 5-phosphate into ribose 5-phosphate with an isomerase. After that, it can be interconverted into intermediates of glycolysis and gluconeogenesis, so any excess of the ribose 5-phosphate can go through glycolysis. This will allow the cells to adapt sugar metabolism to their needs. There's going to be no NADPH generated or consumed. So this first reaction is reversible, as are all the ones that follow from here. Isomerases are enzymes that change things around within the molecule. We're not going to be losing anything here. So our ribulose 5-phosphate isomerase is the enzyme that catalyzes this reaction, where we go from the ribulose 5-phosphate to ribose 5-phosphate, which is still a 5-carbon molecule. A second enzyme can come in with the ribulose 5-phosphate called ribulose 5-phosphate free epimerase, generate xylulose 5-phosphate, which still has 5 carbons. These two reactions here can happen in parallel, with some of the ribulose 5-phosphate going to ribose 5-phosphate and some going to xylulose 5-phosphate. The xylulose 5-phosphate and ribose 5-phosphate generate glyceraldehyde 3-phosphate that has 3 carbons, and cetoheptulose 7-phosphate, which has 7 carbons. With the enzyme Transketolase. The Transketolase transfers a 2-carbon unit from the xylulose 5-phosphate to ribose 5-phosphate, giving us that 7-carbon cetoheptulose 7-phosphate, being left with the glyceraldehyde 3-phosphate. So our transketolase is the enzyme transferring that 2-carbon unit from a ketose donor to an aldose acceptor. This transketolase will need thiamine pyrophosphate, or TPP, as an essential cofactor for this reaction to occur. A transaldolase then catalyzes the transfer of 3-carbon unit from the 7-carbon S7P, which goes to the 3-carbon glyceraldehyde 3-phosphate, to give fructose 6-phosphate, which has 6 carbons, and erythrose 4-phosphate, which is 4 carbons. And this fructose 6 phosphate, as well as that previous glyceraldehyde 3 phosphate, can both be shunted off to other pathways like glycolysis. With another transketolase and bringing in one of the xylulose 5 phosphates with that erythrose 4 phosphate, we get glyceraldehyde 3 phosphate and fructose 6 phosphate, which again can both enter glycolysis. The transketolase will again need its support TPP, and here it's the xylulose donating those 2 carbons to the erythrose 4 phosphate, forming glyceraldehyde 3 phosphate and the fructose 6 phosphate. If we zoom out a bit and look at all of the names and how many carbons each has in the non-oxidative phase, knowing what we know about the structure, we can see that the phosphate is always on the last carbon. The total number of carbons for each of these lines of the reaction is going to be 10, 10, 10, and 9 after adding. So we'll have 5, 5, 7, 3, 6, 4, 6, 3. Our first and last transketolase moves the 2 carbons, while the transaldolase moves the 3. So to remember, to start, 5 and 5 makes 10, shift 3, still 10, shift 2, 1 less, gives us our 9. Or if you remember pathways better with stories, we can have ribose and xylulose walk into a bar. Xylulose lends 2 carbons to ribose for drinks. Ketoheptulose gets into a fight with glyceraldehyde 3 phosphate, losing 3 carbon teeth. Erythrose calls xylulose for backup, and xylulose throws 2 punches. The pentose phosphate pathway is primarily regulated by NADP+ levels. Since glucose 6 phosphate is used by both this pathway and glycolysis, and both take place in the cytosol, there will be shifts in which pathway uses more of the glucose 6 phosphate pool. First enzyme here uses NADP+, so low concentrations of NADP+ will slow this step down. Cells that are rapidly dividing will use a lot of NADPH, so concentrations of NADP+ will rise, stimulating glucose 6 phosphate dehydrogenase and giving us more flux through this pathway. NADPH is also an inhibitor, and it will allosterically bind to glucose 6 phosphate dehydrogenase to further inhibit it. When the NADPH levels are high, there's going to be no need for the oxidative portion. So if the cell doesn't need a lot of NADPH at the time, more of that glucose 6 phosphate will go through glycolysis. The opposite is also true. If the cell needs more NADPH, glucose 6 phosphate will go through this pathway. And because of that one pool for our starting molecule, pathway utilized depends on the cell's need for ribose 5 phosphate, NADPH, and ATP. And there's 4 general scenarios for this. The first is when the need for ribose 5 phosphate is greater than the need for NADPH. This can happen in rapidly dividing cells synthesizing a lot of DNA. Most of that glucose 6 phosphate will go through glycolysis until we reach the fructose 6 phosphate and glyceraldehyde 3 phosphate, both of which will then go through the non-oxidative phase of pentose phosphate pathway and reverse up to the ribose 5 phosphate without generating NADPH. So when we have a greater need for ribose 5 phosphate than we have for NADPH, We get a reverse of the non-oxidative phase from glycolytic intermediates. The second is when our need for NADPH and ribose 5-phosphate are the same, and the cell's needs are balanced. For this one, we'll go through the pentose phosphate pathway, and there's no need for the glycolytic intermediates or additional cycling. Number 3 is when our need for NADPH is greater than that of ribose 5-phosphate. This can happen in the liver and adipose tissue, where NADPH is needed for fatty acid and cholesterol synthesis. We'll have that glucose 6-phosphate go to ribulose 5-phosphate by the oxidative phase of the pentose phosphate pathway to get that NADPH and ribulose 5-phosphate. The ribulose 5-phosphate will then go to ribose 5-phosphate and continue on until we get glyceraldehyde 3-phosphate and fructose 6-phosphate, which can go through gluconeogenesis to get more glucose 6-phosphate that will then go through the oxidative phase of the pentose phosphate pathway, repeating this cycle. With this, as a theoretical maximum when all carbons are oxidized, every glucose 6-phosphate will produce 6 CO2 and 12 NADPH. So when our need for NADPH is greater than that of ribose 5-phosphate, we'll have things going through the oxidative phase with recycling through gluconeogenesis. And number 4 is when we require both NADPH and ATP. So our glucose 6-phosphate is going to go through the oxidative phase of the pentose phosphate pathway to get NADPH. The ribulose 5-phosphate will then continue on until it gets to the fructose 6-phosphate and glyceraldehyde 3-phosphate in the non-oxidative phase to then go through to glycolysis to get pyruvate and ATP. With the pyruvate being able to go through the citric acid cycle to produce more ATP. So when the cell requires both NADPH and ATP, we have a mix of the pentose phosphate pathway and glycolysis. There are a number of conditions that can result from the deficiencies in some of the enzymes used here. Our first enzyme in the pathway is that glucose 6-phosphate dehydrogenase. It has an X-linked condition that leads to glucose 6-phosphate dehydrogenase deficiency. And since this first step is to produce NADPH is diminished in that its level activity is reduced, we'll have more of that NADP plus buildup. When that happens, this results in oxidative damage that eventually leads to the breakdown of red blood cell membranes and the release of free hemoglobin. So they'll have impaired glutathione production that results in oxidative damage and hemolysis. Most will be asymptomatic unless or until they ingest certain foods or medications. One of those foods is the fapabine, and it has been known to cause favism since ancient times. Now there is a genetic basis for this, and it is geographically located in highest levels in certain areas of Africa and the Mediterranean, in areas where malaria is common, and this glucose 6-phosphate dehydrogenase deficiency is associated with a level of malarial resistance. Malaria uses the NADPH in the cell, increasing oxidative stress that can't be readily combated, which leads to both the cell and the parasite dying. Since the parasite can be killed by oxidative damage and stress, this is a similar mechanism that one of the malarial drugs used as a treatment for malaria uses. Or Transketolase and Transaldolase. These are both enzymes that are unique to this pathway. The transketolase reaction required TPP as a cofactor, which is a derivative of vitamin B1, which is also called thiamine. And a lack of thiamine can lead to two notable conditions, beriberi and Wernicke-Korsakoff syndrome. These are primarily thiamine deficiency diseases that impairs transketolase, and transketolase mutations can be an aggravating factor. Beriberi can lead to swelling as a result of accumulating body fluids, paralysis, and even fatal if left untreated. While Wernicke-Korsakoff syndrome causes issues with voluntary movements, and this can include abnormal eye movements, your walking movements, and neurological defects. This is more common in heavy drinkers because heavy drinking interferes with the absorption of thiamine in the intestine. A mutation in the transketolase enzyme results in a lowered affinity for that TPP, making these individuals much more susceptible with worse symptoms. For Transaldolase, there is a very, very rare deficiency associated with it. It's an autosomal recessive, so those impacted will receive one copy from each parent. Because of Transaldolase's location in the pathway, the deficiency will result in high levels of acetylheptulose 7-phosphate. Clinically, it causes diseases of the liver, skin, blood, and dysmorphic features. It was relatively recently discovered, and since it is so rare, and there isn't any known treatment for it yet. So that's the pentose phosphate pathway, where we go from glucose 6-phosphate to ribulose 5-phosphate to get that NADPH, with the ribulose 5-phosphate then going through further reactions to generate those glycolytic intermediates that can then be used for ATP generation or regeneration of glucose 6-phosphate for continued NADPH production.